Okay, our first speaker this afternoon is Georgianna Silveira. She is going to give us an update on the New York Mammal Survey. Okay, Georgianna? Down arrow advances, up arrow goes backwards, and then start your raise your palm.
Thank you so much. All right, thank you so much for the introduction, and thank you for having me here today to just give a little update about the New York Mammal Survey. Yes. Okay, before we get into things, I just wanted to start off by acknowledging some of the individuals and organizations who have made this, as you will see, massive statewide project really possible. Our work would not have been able to be as comprehensive as it is without the folks listed here, including all of the field crews and the students at the SUNY College of Environmental Science and Forestry who helped us out with a lot of the field work and data processing. So the main objective of this New York Mammal Survey is to complete the first coordinated statewide assessment of the status and distribution of all terrestrial and freshwater mammal species in New York State. So basically any mammals except for the ones living in New York's ocean waters. Our goals with this project are really twofold. First of all, to provide information to the State Department of Environmental Conservation on current mammal distributions in order to inform their active management actions, as well as to provide a baseline of mammal distribution against which future studies can be compared. Part of this project has been collecting new data through our statewide trapping efforts, as well as collecting and organizing all of the existing data that's already present in the state on our mammals, including databases collected by the state agencies, public records of mammals on iNaturalist, an extensive review of historic literature, and data provided to us by other researchers and collaborators in the state. Today, I'm mostly just going to focus on our new data collection, our statewide trapping efforts, but I'm happy to take any questions or answer anything about any of these other sources as well as we go through it. So to start off, we, when thinking about Mammal Survey, organizing our field efforts, organizing our literature collection efforts, came up with a list of 66 native species of New York. These are mammal species that either currently exist in New York State or once had established populations in the state. We also identified 6 non-native species. I don't expect you to read this whole list of text. This is just going to be a demonstration for our data collection efforts, but you're welcome to go through and read if you're interested. These are the species for which our eventual reporting to the DEC will include. So we're going to write about the history of these species in the state, their current occurrence, if they do occur, et cetera. So these are sort of all of our target species. I'm going to focus here mainly on the list of the natives. We started off by paring down this list to only the species that still currently exist in New York State. So we have 7 species that were once found here, but still are not. So obviously we can't collect any new data on them. We also can remove from this list 30 species for which sufficient data on their occurrence in New York State basically already exists. These include species that have current or recent research projects measuring their occurrence in the state. These include fishers, snowshoe hare, American black bear, things like that. It also includes species that are sort of actively monitored by the state, including harvested species or species that have health concerns. So we already kind of know a lot about their distribution. So we didn't have to spend a lot of time collecting new data, and instead focus our efforts on this list of 29 species for which we're still not really sure where they exist in New York. You need to get some more information or some more detailed information about where to find them. And you'll notice that a lot of them are the very small guys, which definitely requires a lot more effort to measure small animals at the statewide scale. So I'm going to describe some of our many data collection methods. We've included a lot of different trap types for this whole project, which our field work started for the Mammal Survey in 2019. So we've been going strong for 6 years now. And we started off with our standard trapping survey units, which mainly consisted of the 3 types of live traps that you see on the slide here. So these traps are all designed to catch animals alive. Our survey units consisted of 150 traps at each unit, and a unit consists of that set of traps set out at the same time in the same sort of homogeneous habitat type. And so we can measure the species occurrence at that like patch of habitat using our 150 traps of different types. And so I just listed the numbers here for how many of each trap we set out across the state, so you can see how much effort was put into this into this project. All the traps were set out for 6 total trap nights, which are a trap night is considered an overnight period where the trap was open and able to be catching animals. We had them set out on a 4 nights closed, 3 nights open, 4 nights closed, 3 nights open schedule. So 2 total weeks of trapping. When animals were captured in our traps, we collected a lot of information about like what species they were, obviously, but also took measurements, especially like measurements on reproductive status and other health effects. And we also, when necessary, took tissue samples, generally in the form of an ear punch, where we cut a little tissue off the ear, which we can bring back to the lab for genetic analysis. If an animal was found dead in our traps, which does unfortunately happen, then they were taken also back to the lab to become part of our research and teaching collections at SUNY ESF, which I will talk a little bit more about later. And also a subset of animals were humanely euthanized, again, just to become part of our research and teaching collections and sort of fill out that data set. As well as those 3 types of live traps that I showed you, we also had a few additional traps that were set out as part of these standard survey units when the conditions were right, when the habitat was right, when we sort of needed that data. First, we set out 2 types of trail cameras. One set to try to capture flying squirrels and one to try to capture weasels. The flying squirrel cameras were baited with like peanut butter, seeds, and oats, and the weasel cameras were baited with meat, which definitely brought along some unwelcome visitors, as you can see. We also set out acoustic recorders across the state, again, at some of these standard survey units to collect flying squirrel calls. Starting in 2023, we started implementing a new survey protocol that was aimed to detect species for which we really hadn't collected a lot of data using our standard survey units. So these units were set out, the standard survey units were set out mostly on state-owned property, whereas these targeted survey units were placed out in habitats where we expected to see some of these species that were a little bit more rare, a little bit more cryptic in our survey efforts. Namely, they were set out at like marshes, bogs, and taluses, which you can see there. These targeted survey units consisted of less invasive survey methods. Specifically, we set out 2 types of bucket cameras, bonk and bonk, which are just like 7-gallon buckets with trail cameras affixed to the top. They point down at the bottom of the bucket with holes in the side, and you can see the little guys running in and out, which is a sort of newer method for being able to harness the like surveying power of trail cameras to detect small critters. We also, in these targeted survey units, set out seed trays, which are actually modified rodenticide bait containers. But instead of putting lethal baits in them, we just baited them with seeds and mealworms and used these seed trays to collect traces of DNA called environmental DNA that we can sequence and use to identify species at these survey sites. So our targeted survey units were set out for 4 total weeks. Up to 2 bucket cameras and up to 2 seed trays were set out at each survey site, again, in sort of a homogeneous habitat. Set out for 4 weeks, set up once and checked twice. So significantly less effort for our field crews, which I'm sure they appreciated. Okay, here is a map showing our total surveying effort across the state. Each point on this map doesn't represent an individual trap. It represents a individual survey unit, so sort of like the central location of all the traps that were set out at each time, color-coded by the protocol type that was followed at each unit. And I just really want to highlight again how much effort this all was and how cool it's been to be able to get this much spatial coverage across the state. We've covered a lot of different habitat types and been able to set out a lot of cool and interesting types of traps to try to capture all of New York's diverse mammals. And because we are on Long Island, I just wanted to highlight some of the survey effort done on Long Island. Mike Bettini's talk later will go into some more detail on some of the Long Island data, but I did just want to highlight all of the effort that's been done here. And I think, isn't this where we are? So fun. No? Oh, like here-ish, which is kind of fun. Great. And so I just want to really highlight the efforts done by the CTEC staff, by the Nature Conservancy staff, and the Region 1 and 2 biologists on the DC who were able to accomplish all of this survey effort on Long Island specifically. So now that we have all of this data in hand, we've been collecting data since 2019 out in the field. What are we then going to do with it? We have to try to figure out which species are at all of these sites from all of our methods. So there's a couple, I would say 4 main ways that we use our data to identify the species that are present at all of our survey sites. First and easiest, of course, is just identification in the field. There are a number of species that are very easy to identify by the naked eye. And so most often in these cases, field crews just noted down what the animals were and let them go, basically, which they were probably happy about. And so there's some species, right, like you can see a star-nosed mole here. We've got a skunk, some things that are sort of easy to identify by the naked eye. But of course, there are some, especially some of the very small animals that we cannot identify to the species level just in hand in the field. So we have some other methods to try to figure out what these guys are after the fact. Same thing with our trail camera photos. So here are some example photos from our squirrel and weasel setups, where we got some of our target species, some non-target species, all very interesting and silly pictures from across the state of some of our mammals. And again, just going through these, identifying them to the species or sometimes genus level whenever possible to process all of these photos. Same thing with our trail camera bucket photos. I personally am currently working with a team of 22 undergraduate students at SUNY ESF who have so far this semester identified species in around 1 million of the photos that we've collected from bucket cameras, and we still have around 1 million to go. So I've been very appreciative of all of their hard work, figuring out what we're finding in all of the buckets. While it is mostly mice, they're having a fun time in there. We also have been seeing some examples of the target species that we're looking for, like shrews and voles, as well as some surprise guests, such as this fox that was able to stick its head into the bucket somehow, this cool snake, and a lot of sneaky raccoon hands trying to steal our bait out of the buckets. So those are sort of our main, like, in the field identification techniques. When voucher specimens, so like, deceased animals, were taken back to the lab, there are ways to more closely examine these specimens and be able to identify to the species level. So you'll see one of our principal investigators on the project, wrong one, observing all of the many vouchers that were taken from the field in their fancy Ziploc bags, as well as our efforts to, really try to peruse the details of these animals and figure out what the species are. Some of them, especially the really small ones, you have to like open the mouth and count their teeth to like get identification. So it's kind of a laborious process. And then these 2 photos here are not actually specimens that were taken from the mammal survey, but they are photos from the Roosevelt Wildlife Station Natural History Collection housed at ESF, which is going to be the eventual home of many of these samples taken. So I just wanted to show what sort of like storage we have for these types of specimens and their eventual use in our research and teaching collections at the university. Our final and most complicated, I would say, method for species identification is using genetics. So we collected 2 types of genetic data for this project. We collected genetic material directly from animals, generally taking, like I said, an ear clip. So like taking a tissue sample, we can bring that back to the lab, sequence it, and be able to tell what species it is, which is especially helpful in the case of our 2 species of mice in New York. Paramyscus. And then we also, like I said, collected the environmental DNA. And so we use these these sort of like paper swabs, and you can just like swipe the inside of our seed trays as well as the bucket cameras and pick up traces of DNA that are shed by animals, like as they're living their lives and eating and peeing in our traps and all that. You can collect that genetic material and bring it back to the lab and sequence it, and at least in theory, identify which species have been present at that site, which is a really useful and like kind of cool, non-invasive method for monitoring these animals. So we have basically all of the tissue species IDs completed for this project, and we are like in the final stages of starting to get our environmental DNA results out, which are very exciting. So going back to our list of 29 species, I just want to highlight who we have found so far, which is 23 of our species, which I think is pretty good. I will just like put out a caveat there that everything is not finalized yet. This is the best of our knowledge so far, so we can't definitively say either way whether these animals do or do not exist in New York State. We're still waiting on our environmental DNA results, still doing our final data cleanup push, our final processing of the public data from my naturalist, all of that. But so far we've detected 23 of our 29 target species. Across all of those methods that I mentioned, our live trapping, our camera trapping, our tissue like collection, basically all of our methods besides the environmental DNA, we've had 34,609 detections of mammals across New York State. 69% of which have been mice. They are definitely dominant in all of our survey sites. We've collected a lot of mouse data. But I do want to highlight some of the interesting detections that we've found throughout this project. Like I said, we're still processing through especially some of the more rare species data, but here's what we found so far. We were very excited to find a detection of a species that we had been searching for basically all of 2024 field season, which was the least shrew. This is one of the smallest mammals in the world, and it was detected on Long Island on Orient Point. Rediscovered in the state for the first time in like around 100 years. We had some historical records of least shrew from like the mid 1930s, and we're super excited to see it on Orient Point. So the only evidence that we have of it so far is this one photo, which has been looked at, and like many experts that we've talked to agree that this is a least shrew, but we are doing some additional surveying out on Orient Point to try to get more detections of this animal potentially collect a voucher to really confirm that it is actually present in the state. But we're very excited. Another interesting detection that I wanted to highlight was our detection of the long-tailed shrew. So we have 2 detections of this animal in New York, as well as, again, some historic records from like the 1930s to the 1990s. Those detections sort of span a couple of decades. But we've had one confirmed detection in our bucket cameras, which is the black and white photo that you can see, and one voucher that was identified to be a long-tailed shrew, although looking at it again, I'm not sure that the tail is long enough, but it's labeled as long-tailed shrew for now, so I included it in here. But something that's kind of exciting about this specific long-tailed shrew photo is it was suggested by one of our colleagues to be one of the only photos taken of a living long-tailed shrew. Shrews don't really survive very well in traps. Their metabolism can't really handle it. So often they just like die even in live traps. And so it's kind of rare to see them sort of living in general. And this photo specifically was exciting because it's just like a long-tailed shrew living its life, which shows one of the cool aspects of the bucket photos that I think make this surveying method kind of unique. So I also wanted to just show some representations of how we might be thinking about displaying this data or summarizing it for our final reports. For example, this is a county level summary of our jumping mice detections across all of our surveying efforts, highlighting counties where both species occurred, one or the other, as well as some counties with unidentified species that we are hoping will be filled in by the eDNA results when those come in. And then this is a historic versus current comparison of another very commonly detected species. Besides mice, the short-tailed shrew was our second most commonly detected species in the state. And just like a historic versus modern comparison, like comparing those literature records that we looked at to our trapping results, which I expect this to be more filled in as well especially once those eDNA results come in, just because they are so prevalent across the state. But here's another example of the ways that we can summarize and sort of look at this extensive data set. So what's next for the mammal survey? Our project is slated to end in spring of 2026. So we have like around a year left. Basically, all of our field efforts have wrapped up at this point. We have all of our data together and are in the process of doing our species occurrence maps and final data cleanup. Our plan is to produce two types of reports summarizing our data. One is a more technical report for the DEC to, again, inform their management decisions and understanding of mammals in the state. And one is a more public-facing report for the scientific community and interested public that can provide a record of this project and our findings on New York mammals. So those are scheduled to come out in around a year. So, keep you posted. And that's actually all I had. I wanted to make sure we had time for lots of questions, if anyone has them. So that's all I have for you all today. Thank you so much. Are there any fishers on Long Island? No, not from what we found so far. I don't think so.
So I have a couple questions. I could skip over in my presentation. So when you did the tissue samples, were you finding any deer mice on Long Island, or was it all white-footed? Do you recall that?
I don't know. And so we, like I said, collected like a lot of mice and didn't genetically identify all of them. I think we just did like a subset to get the species. I think, again, the environmental DNA would like elucidate that further, but I am not sure about the like differences just yet. Yeah. Oh. Yeah. Oh, that's a weasel eating a vole. So this is from one of our bucket setups. So the camera is like above it, pointing down at the ground. And so this animal is like looking up, holding a vole in its mouth.
Yes,
Yes.
we do have weasels on there. And that reminds me of another question. Did you confirm any short-tailed weasels on Long Island?
Short-tailed weasels. I think I don't even know what I just did. I think so far. Right. So I know, so far we haven't confirmed, I haven't confirmed from the data that I've looked at and like done cleaning and verification on. I haven't seen any short-tailed weasels just yet. The weasel work is a little bit more intensive, and some of the other people on the project are doing like a little bit of extra effort to sort of like clean up those like weasel species identification methods from trail camera photos. And so that work is like coming out very soon. But so far, like confirmed to the extent that I was like comfortable sharing today. I haven't seen any confirmed short-tailed weasels, but like I said, we're still in the early processes of data cleaning.
Then, did you do any trapping for moles in the Corals River? Do you recall?
I don't
That's
think
where
so.
we had Connor found the star nose.
Right. I don't think we've done a ton of mole trapping specifically, and because I think we were planning mostly to source our mole data from the state records on like nuisance wildlife reports or like pest reporting. So we haven't done a lot of specific trapping for moles.
I have an annoying question about,
Oh, yeah.
the overnight traffic.
Sure.
For the pit bulls.
Right.
Were those at the same kind of time frame as like the Havahars and the Shermans, or was there a compressed frame like 3 or 6 hours instead of the full overnight?
Oh, for the pitfall trapping, I think they only went out once a day. I don't like entirely recall. By the time I came on, the pitfall trapping had mostly ceased. And we were focusing more on like the
The bucket.
yeah, the bucket cameras and everything. But I think they can only go out like once a day at most, just because this was a really intensive trapping schedule, unfortunately. Yeah, good question. Thanks.
Thanks,
Cool. Thanks, everyone.